This is Jane. Jane's been seeing her primary care provider for some genetic testing for sickle cell anemia, which runs in her family. Displayed here is a pedigree of her family, showing what relatives are affected by the sickle cell gene mutation according to genetic testing they've received. Sickle cell anemia is a recessive gene, which means that even though Jane shows no signs or symptoms of sickle cell anemia, she may still have a gene mutation that can be passed down to any kid she may have without knowing it. Because of this, Jane's doctor suggested that she get genetic testing done to see if she's a carrier. Sickle cell anemia is a point mutation, which means a single nucleotide is changed in a genetic sequence and can change the protein the genes encode for. In this case, it changes the hemoglobin protein in red blood cells. While there are many different ways to try and figure out if someone has a point mutation, Jane's doctor recommended gene sequencing, which is able to detect a single nucleotide mutation. To sequence DNA, we first need to get a sample we can extract DNA from. Once the DNA is extracted and purified, we can begin the process of sequencing. Automated fluorescent sequencing is something we use to determine a genetic code by nucleotide sequence. Specifically, it adds fluorescent dye markers to differentiate nucleotides. The dyed DNA is then ran underneath a laser, which causes the dye to fluoresce so it can be read. Unlike other types of genetic sequencing, the reading is entirely automated and will give us a nucleotide sequence automatically. There are two types of automated fluorescent sequencing, dye primer and dye terminator. Once we have our purified DNA, we need to figure out its nucleotide sequence. To do this, we do the steps of DNA replication with a few important changes. We use a primer that's specific to the area we want to replicate. We include dye to differentiate nucleotides, and we include some dideoxynucleotides, which we'll call ddNTP, instead of deoxynucleotides, or DNTPs. ddNTPs don't include a chemical bonding site that connects nucleic acids, which stops transcription on that chain of DNA, unlike the normal DNTP. This is what allows us to sort and sequence DNA. When sequencing DNA, we start with a classic double-stranded piece of DNA. After applying some heat, it's denatured or unwound into single-stranded DNA, so we can replicate it. Primer will attach and start replicating DNA, adding on DNTPs or ddNTPs. Once we've ran this a few times and created a lot of DNA, it will be put into gel electrophoresis, which will sort them by size. Because of how the DNA replication was terminated, we're able to read the gel from bottom, or the smallest fragment, to top, or the largest fragment, and can see how the DNA is organized. To figure out the sequence from the DNA organization, we need a way to differentiate which fragments mean what nucleotides. We do this with fluorescent dyes. We can add the dye in two different ways, to the primer, or to the ddNTP or dye primer and dye terminator, respectively. If we add dye to the primer, we use 4 different tubes, each with a different ddNTP and different dye. This means each tube will have a sample of DNA that indicates where each spot a specific base in a sequence is. We'll add all 4 of these to a gel plate, and after they migrate they'll be read accordingly. The other option is adding the dye at the end to the ddNTP. The main difference is this can be done in 1 tube instead of 4. In dye terminator sequencing, the dye is attached directly onto the end nucleotide already, so we can put all 4 dyes and ddNTPs into 1 tube. After doing this, we also put the sample on a gel plate and it can be read by the dye color and order of the bands.